Good afternoon, good evening, everybody. Welcome to this ATS Journal Club from the Pulmonary Circulation. And I'm quite happy to announce the presentation of the late publication of Ke Yuan in the EMBOJ journal titled Specialized Pericyte Subtypes in the Pulmonary Capillaries. We are very happy to have Ke here at this night. She will, or this afternoon, and she will present the work contained in this publication. I think one very nice issue is that this publication highlights an identification of 2 pericyte subtypes in the pulmonary circulation. And I think that answers a long-standing conundrum in how pericytes may either contribute or protect, or either contribute to disease or at the same time are essential to the integrity of the pulmonary circulation. So I'm very happy to have her here, and she will be cited by 2 international experts in the field of translational research in the pulmonary circulation. The first is Professor Lan Zhao from Imperial College London, and the other is Professor Grazyna Kwapiszewska from Graz University in Austria. And I will be the discussion, or I will lead the discussion today. My name is Jurjan Aman. I'm from Amsterdam University Medical Center in the Netherlands. 1 or 2 points of the logistics for today. During the presentation and also during the discussion, you're allowed to ask questions in the chat, and I'll bring up these questions later on after the presentation. And directly after the presentation, the 2 discussants have the first try to ask questions to the presenter. So I welcome you to address all your questions in the chat, and I'll bring these up in the discussion. So this is it as for the introduction. Ke, I'm happy to have you here, and the floor is yours.
Thank you so much, Yuyan, for your kind introduction. And thank you, Grazyna and Lan, for being here, stay late. Thanks, ATS, for the setting up the meeting. Good morning, good evening for everybody here. Welcome to the PC Journal Club. Today, I'd like to talk about the work we recently published in EMBOJ journal, very hot off the plate. The title is Specialized Pericyte Subtypes in the Pulmonary Capillaries. I would like to thank all the co-authors and co-first authors and co-corresponding authors contributing to the work. So I'd like to begin with some quick introduction about the cell types we have been dedicated to study. It's called pericytes. They are mesenchymal-derived mural cells that wrap around endothelial cells or capillaries throughout the body. They are embedded with the basement membrane, which direct contact with capillary endothelial cells. Their presence are required for capillary stabilization, maturation, and remodeling. So my lab has been spent a lot of time to study these controversial cell types and how to distinguish them from mural smooth muscle cells. Pericytes are located in capillary beds, which means the vessel diameter is less than 10 micron size. They have oval cell body with long processes, and we use 3G5 PDGF receptor beta for staining them, and they're negative for smooth muscle actin. Whereas smooth muscle cells, they're spindle shape, and they're located in arterioles and arteries, and they are positive for smooth muscle actin and myosin heavy chain 11. So to date, there's no single markers can exclusively identify a pericyte and distinguish them from other cell types. Therefore, characterizing the role of pericyte in disease development remain very challenging. In my previous studies, we use CSPG4 CRE line to bred with TD tomato. Therefore, we can label them in capillaries. As I shown here in this landscape of the vasculature here, smooth muscle actin definitely stain the bigger size vessel, whereas TDT cells, which are CSPG4 positive, CRE positive cells, you can see they label capillary pericytes very well. Unfortunately, there are some TDT cells in the arteries, which are spindle shape indicated by this white arrowhead, indicating CSPG4 also labels smooth muscle cells. The same issue applied to PDGF receptor beta CRE lines, which I show two of them we have been worked in the past. As you show in here, this is a outline of a artery. You can see the TD tomato cells can label both pericyte and smooth muscle cells. So in order to identify a unique pericyte marker, my talented postdoctoral fellow Han looked into both single-cell RNA-seq data for human lung and mouse lung publicly database. And according to the classic markers of CSPG4 and the PDGF receptor beta, we are able to identify pericyte clusters in these database. And he applied DEG analysis in these pericyte population in both lung and heart organs. We identified the significantly upregulated genes. So here in this table, you can see there are two significantly upregulated genes, and one of them is HIGD1B. See? Here we go. And sorry, it's a little bit slow when I try. Here we go. And in the mouse lung, HIG1B is the top upregulated genes. And in human heart single-cell RNA-seq data, we don't see any gene met the criteria we're looking for. And in the mouse heart single-cell data, HIG1B is the only one significantly upregulated genes. So therefore, we're interested in this HIG1B gene. So we just look more closer in the human lung single-cell data based from the human lung cell atlas database available in the Chan-Dagerberg initiatives. So from the left, that's the dot plots. And the violin plots simply validate these dot plots data. You can see we use the three classic pericyte markers, CSPG4 and PGG receptor beta, and HIG1B showing this is the circle seem to be only specific to the lung pericyte, although we see a very small expression of level of smooth muscle cells as shown here. And the smooth muscle markers in these data sets, we use ATAC2, CNN1, and TAGLN. And then we look into the UMAP of the human lung single cell data. And here showing this is pericyte cluster, and there's smooth muscle cell cluster. And for each genes, we also use density plot to show the expression level. So when we zoom in at these two populations here, you can see HIG1B seem to be very specific to the area that co-expressed by PDGF receptor beta and CSPG4, whereas the rest of the three seem to not co-localized well with pericyte markers. Then we look into the Tabula Muris database here, showing the UMAP here showing the sample UMAP, and you can see pericyte population here in this kind of orange color, and the pulmonary arterial smooth muscles are in the area above it. And here, this kind of purple-like color is bronchial smooth muscle cells. So using the same density plot, we can also see HIG1B seem to be only label that area that suggesting these pericytes, whereas the other 3 markers for smooth muscle cells didn't label pericyte well, and they specific label the pulmonary arterial smooth muscle cells. Then, based on the single cell data, we decided to validate the HIG1B expression levels. So we first used the RNA-scope to detect its expression levels in CSPG4 TT tomato mouse OCT slices. So you could see the TT tomato positive cells are also positive for HIG1B. And HIG1B in this zoom-in image seem to label more cells, like the one at the lower corner here, indicating HIG1B could label more pericyte than the NG2 expression cells. So after these single cell data and RNA scope data, we collaborated Mantu, who is the director for the Harvard Mouse Genome Core, and he used CRISPR knock-in techniques to creating this HIG CRE-ERT2 animals for us. Then we bred these animals with the reporter line, which is AI14. And this is the work carried on by Tim and Renee, who are both very talented fellows in my lab. And for here, the figure A, the panel A here, showing the TDT are in the parenchyma, smooth muscle actin, labels arterioles, and the TDT are negative for CD31. And then we also conduct the antibody staining for these lungs. By the way, this is position cut lung slices. So these images are in 3D. And here, the green is showing PDGF receptor beta labels almost 100% TDT cells. When we use NG2 antibody, it's also labeled TDT cells very well. The quantification are shown in the panel C. And we also label these lung sizes using other markers for MHC2, CD11C. We use LY6G for neutrophil markers and CD326 for epithelial markers. TDT are all negative for these markers. To further validate the CRE expression efficacy, we bred HIG1B mice with MTMG reporter, and showing here, the GFP clearly label pericytes in the capillary level, and they are negative for smooth muscle actin, and they are negative for CD31. They are positive for PDGF receptor beta. We are also interested in pericyte in different organs, particularly in heart. We are able to show HIG1B TDT also label pericyte in heart very well without labeling neural cells, smooth muscle cells, as shown here in smooth muscle actin. This is basically one of the questions from reviewers. What about HIG1B? Does it label other pericyte in different organs? So my research assistant, Yan, carried on this staining studies by collecting different organs in the HIG TDT mice. As you shown here, TDT labels pericytes in brain, in muscle, pancreas, heart, intestine, descending, the connective tissue around descending aorta, and very well labeled in kidney and retina. However, we don't see any TDT label in liver or spleen, suggesting HIG1B can be labeling pericyte in an organ-specific manner. So in lineage tracing, we all know the tamoxifen can be off-target effect. So we also conduct many negative control experiments. So for here, without CRE, we also at the mean tamoxifen, we don't see any TDT being labeled, or in MTMG line, any GFP being labeled. And without tamoxifen injection, each HIG1B TDT or MTMG reporter didn't have any reporter color label. That means our CRE line didn't have any leakage at all. So after all this validation, we're very excited to have this transgenic knocking line can precisely label pericyte in the lung. We want to study how pericyte contributing to the development of hypoxia-induced pulmonary hypertension. So we subject the mice under different time points of hypoxia, as here in 1 week, 2 weeks, 3 weeks exposure of 10% oxygen hypoxia. And we collect the tissue and conduct immunostaining. So pneumoxia, we see pericytes are in capillary levels. And after 1 week, we see some of the cells wrapping around the arteriole and become small positive. And there's more TDT cell coverage of the arteriole at 2 weeks, and as well as in 3 weeks, where the pulmonary hypertension will be established after 3 weeks of hypoxia. Then we got another question is how to quantify the number of the TDT cells under 3-week hypoxia. What about their coverage in veins? So we also conduct the prediction cut lung slices. So on the left, you could see the anatomy of the mouse vasculature. Here, the BR represents the label of the bronchial, and arteries in the letter A represents arteries that is parallel to the bronchial. And in between the two big bronchioles, usually there is a vein. And another way to tell apart artery and vein is the pattern of the smooth muscle actin staining. And usually the vein have a reduced intensity of smooth muscle actin staining. And here we're showing the position cut lung slices after 3 weeks of hypoxia. And we have zoom in of this vein, 2 of the veins, as well as the artery. And we quantify how many cells covering artery and vein, and showing the graph here. Basically, we don't see much of the TDT coverage in the vein, but after 3 weeks of hypoxia, we do see pericyte increase like 10% coverage in the artery, and there's reduction of pericyte number at the capillary level. One of another interesting phenomenon we found is there's dynamic pericyte movement. So as we know, we subject black 6 mice under hypoxia, we can establish pulmonary hypertension. And an interesting phenotype we also being found, also published in the prior literature, is if we move these animals back to pneumoxia for another 3 weeks, we call recovery model, these mice are completely recovered from pH. Their hemodynamic level are completely normal. So with that, we also conduct lineage tracing for the recovery model. So TDT here, I'm showing they're covered around the arteriole, they co-express smooth muscle actin, and after recovery, we don't see much minimum, like almost none TDT coverage. A majority of them move back to capillary and negative for smooth muscle actin. So that's very intriguing for us. We don't know why pericyte is moving this way. So we are trying to figure out how they move because of the recovery, and why they want to move this way to maintain the hemostasis of the pulmonary circulation. So go back to the published work is we further look into the pericyte subpopulations. by single-cell RNA-seq data. And here is the human lung cell atlas. We took all the pericyte populations among almost this 24 donor population and reclustered them. And after reclustering, we do find cluster 2 seems to be very different expression levels compared to the others. And among cluster 0 and 3, they all have high level of the pericyte markers. We are just comparing the relative expression level among all this cluster, and we decided to further evaluating the cluster 0 versus cluster number 2, based on the expression density levels of CSPG4 and PTGR receptor beta and HIG1B. So the panel B here showing the cluster 0 have relatively higher expression level of HIG and a relatively lower expression level of PTGR receptor beta compared to the cluster 2. And we define, and also when we look at the KEGG pathway, cluster 0, we think they are type 1, they are classic capillary pericyte. Their pathway is very relevant with hypoxia and reactive oxygen species pathway. And when we look at the type 2 pericyte population, their pathway is highly relevant, like a TGF-beta and regulation of actin cytoskeleton movement and vascular smooth muscle contraction. So that's why we call this class 2 is as lineage active pericytes. Then in order to further study the differences of these two types, we look into publicly available idiopathic pulmonary arterial hypertension single-cell RNA-seq data from GSE database. And we do see cluster number 5 and 6 both express HIG1B, but type 1, we call the cluster 6 as type 1 because the HIG1B expression level is higher than the subcluster number 5. And for the cluster number 5, we call them type 2 because their HIG1B expression level is relatively lower than cluster 6, and their PTGR expression level is higher. So to validate that, we just look into the HIG-TDT hypoxia samples. We see the type 1 cell, type 1 pericyte, which labeled by TDT, they have more processes, and they are more located in capillary nets and far away from arterioles, where we think the type 2, which is lineage active ones, are having shorter processes and they are closer to the arterioles. So after 3-week hypoxia, these type 2 pericyte closer to the arteriole would translocate it to the arteriole and become small positive cells. They also change their morphology, become more flat and lose their processes, whereas type 1 cells, type 1 pericyte remain at the capillary level. So in order to further see the expression level differences between type 1 and type 2, we look into DEG expression levels in type 2 pericytes, and we found Vimentin is more upregulated in type 2 cells. And from a mouse 3-week hypoxia versus control single-cell RNA-seq data, we are able to look into pericyte population and also found Vimentin is significantly upregulated after 3-week hypoxia in pericyte cluster. So therefore, our next validation is, can we look into Vimentin expression levels in type 2 cells? So we further conduct precision cut long slices staining, and Vimentin in grain, we could see in type 2 cells, under normoxic conditions, they're close to the arterial, and they don't express Vimentin. And after 3 weeks hypoxia, these cells closer to the arterial, wrapping around this small positive arterial, and they co-express Vimentin. So here is all the in vivo data. Our next question is, what about Vimentin effect on human pericytes? So we overexpress Vimentin by using the plasmid. And here, the panel A showing, this is adhesion assay. We overexpress these Vimentin in healthy pericyte, and we just seed them on a glass-covered slides. You could see the cell after 24 hours, they cannot spread well. And with hypoxic treatment, it is even worse. These cells just forming this ball shape on the slide could not spread well. However, with Vimentin overexpression pericyte, under normoxic condition, they completely spreading themselves on the slides. With hypoxic condition, they can spread even better, suggesting these Vimentin expression levels under hypoxic stimulation can help this cell spreading, or suggesting they could maybe help with their migration. So that leads us to the migration assay conducted by using wound healing assay. Here you can see after scratch, this is after 6-hour migration, and the cells are stained by F-actin. And here is normoxia after hypoxia conditions. Without Vimentin expression level, hypoxia can stimulate more cell expression, more migration. And with Vimentin overexpression, they almost close up the gap in between. And here on the right side, indicating the number, the distance they migrated. The Vimentin expression under hypoxic condition stimulate pericyte migration. This is our final working model. Pericyte were first described in 1970s. So over the almost 200 years, we only scratched the surface of it, and we only understand very limited biology of it. So there are a lot of more work to done, but we're excited to figure out HIG1B is a unique gene marker for pulmonary pericytes that is more specific than CSPG4 and PTJ receptor beta. These HIG CRE-ERT2 knocking mice will allow accurate fate mapping experiments for pericyte in lung, hearts, and other organs. Then that could help us to understand their basic biology in many disease progression and development. And I want to advocate single-cell RNA-seq analysis in human and murine lungs that also can help us to understand more biology of 2 HIG-positive pericyte subtypes. And lastly, our study show under hypoxia, type 1 pericyte remain quiescent in the capillary network, but they definitely prolong their processes, whereas type 2 cells upregulate Vimentin and other smooth muscle markers, expression levels, and accumulated in arterial, and contributing to the remodeling process, suggesting their important role in the development of pulmonary hypertension, and potentially we can study the function of pericyte and develop new therapeutics to treat human pulmonary arterial hypertension. So with that, I want to thank all the people contributing to the work and my collaborators and all the fundings make all the work possible. And lastly, I would like to thank for your attention to listen to me, and I'm happy to take any questions.
Wow. Thank you very much for this impressive work. A lot of data, and I think I'm quite impressed by the imaging and the very nice views on the microcirculation and the alveolar views of the lungs. Very well done, and my apologies for not mentioning your affiliation in the beginning. I think you put it nicely on the slide, and by seeing your first slide, I realized that I didn't mention it. My apologies for it.
No problem.
Having said that, I would like to open the discussion because I think it gives very nice food for thoughts. And I welcome first the discussants, Lan and Grazyna, to open the discussion.
Right. Congratulations for such a brilliant work. It's truly a discovery of these two types of pericytes. I still remember to this day when I did my PhD, I had this little review. I drew this pericyte and had a curiosity, what do they do? Because there's no tools like what you're saying, you know, what can we do about it, you know, to discover it. So you've applied brilliant tools here using the single cell RNA-seq, then step by step on bench experimental tools from immunostaining, then the mice model, then further immunostaining, then go back to the human. So I have a couple of questions. I want to raise it for discussion rather than directly ask you about it. So first of all, from those single-cell RNA-seq analysis, how do you made it a confirmation, actually, this is a data you can trust and be able to say, you know, this is a cluster of the cells, this is the labeling of the cells, because they are online data, to be honest. They are, of course, is from someone published studies. How do you judge that? What's the power do you apply to be confirmed go forward of this? The second question, of course, is another method you applied for the immunostaining. I saw you have applied some of the immunostaining to the lung sections, co-stand with, let's say, an example of CD31 with TDT. But for me, I see your CD31, it's everywhere. It doesn't seem to be capillary rather than alveolar. That's just a question towards you, you know, as I see the lung structures, especially on your Hig 1B TDT mice lungs. So that's the second question, specifically about the methods, because I have this question from time to time, you know, and you claim actually those TDT isn't in CD31, isn't in airway, but the background immunostaining to me is very, very strong. The other thing is the third question is a pericyte. You did these experiments. How do you isolate it? Was it commercially available to enable you to do all these experiments? Because they are particularly interested to look at the functional side, because you know the hypoxia model, when you see the extension of the smooth muscle cell to the capillary bed, is it pericyte basically migrated or endothelial cells, you know, you mentioned the Vimentin, you know, becomes smooth muscle cell, or is it the old concept is the extension of the smooth muscle cells into the capillary bed? So 3 questions here for discussion, but that's it from me. Thank you.
Well, great. That's all great questions, Lan. Thank you. Well, I definitely put down on my notes. The first question is about the single cell RNA-seq data. before this study, we have published one work with Han, my bioinformatician, when we look into single-cell RNA-seq data in tabula miris. So we look at different organs, like including lung, heart, bladder, urinary bladder, and kidney. And we do find there are about 5 candidate genes for these pericytes. And I don't know which one will be the best one, so I have to test it one by one. And we started with this HIG1B because it's not being widely studied. That means it didn't have a major role in like driving some biology of pericyte. So, but our mice line is a knock-in. It doesn't damage the function of this gene, but because it's not a widely studied gene and it's relatively shorter, so it will be beneficial if we design the CRISPR guide RNA, etc., to lead into the mouse embryo. So another reason is we conduct RNA-scope studies. So that clearly show these punctated mRNA levels of this HIG is co-expressed with NG2-TD T cells. And when we look at the smooth muscle layers, we don't see any of these mRNA expression levels. So so that just gave us some heads up. That could be one potential marker. So that's we just take the risk and work with Mantu and to construct this animal. I didn't mention this work take us about 6 years. Our first batch, somehow the CRISPR didn't work. We didn't get founder mouse. So we have to start all over again. And getting the second batch of the founder mice, we are very lucky. We got 1 founder mice and we are able to cross with TDT. Then we are able to get the precise label. So for the single cell RNA-seq question is, we do find several markers. So HIG is one of them. And the second question is about immunofluorescence staining. And so I don't do any of the paraffin staining in my lab anymore. We did mostly is
yes, this is a slide. Yeah. So I saw the CD31 TDT staining. Particularly interested in this because it seems to me it's all the alveolar, or is it autofluorescence, to be honest? So you do pointed to the probably, there is vessels I could see.
Yeah.
Maybe there is autofluorescence, perhaps.
No, so this is CD31 with the PCAN1 staining. We use AVIA to reconstruct the capillary nets of capillaries. So that's the way how we conduct imaging now. This is not autofluorescence. This is positive staining of capillary nets. And to be published in EMBOJ, I have to provide all the 3D videos, including each single slices. So what I show here is about 50 micron thickness of the long. So in EMBOJ journal, each panel of the images, I have to create a video and upload it.
So this is like the Z series, is it?
This is the stack of confocal images.
Okay. All right. Okay. I understand, because I was thinking is the lung section where you did the immunofluorescence. So, okay. So all your images actually was reconstructed with
confocal. Right.
With a confocal. How many slices were there?
So for these, the thickness is 50 micron, so it's about 50 slices.
I see. Brilliant.
Thanks. So the last question is how to isolate mouse and human pericyte? Or you folks ask for human pericytes?
The human pericytes, you did the experiments.
Yeah. So the human pericytes, we use 3G5 antibody to conjugate with magnetic beads, then isolated them. So I'm happy to share that antibody to anyone who are interested to stem pericyte. That's a ganglioside antibody. Pericyte express abundantly of these ganglioside antigens on the cell surface. So we are able to use that antibody to label them very precisely on human tissue slices, which I don't have it in this work. We published in an earlier work. And that antibody is able to conjugate it with dynabeads. And once the human tissue, we receive healthy lung human tissues from IIAM. It's an organization providing donor lungs. So we can dissociate the lung tissue and incubate with beads and conduct the sorting. And we have been published using that cell pericyte biobank for all of our published work. Anyone who is interested in 3G5 to stain lung tissues, I'm happy to provide it.
Okay. Have you conducted, organelle? That's the other thing people study these days. So mix the cell population study. Let's say you mix the pericyte with the endothelial cell. Did you conduct any of these integrative studies?
So as far as my understanding, if you want to do organoids, you have to do iPSC work. So we haven't done that yet. Those are primary cells. So what we have done is coated some collagen coated beads endothelial cells to forming a little like ball, and we incubate those endothelial coated beads in fibrinogen gel. Then in the matrix, we put in mixture of pericyte. Then the endothelial can form, outgrow these endothelial tubes, and we see pericyte is able to move closer to this tube and enhance the angiogenesis.
Yeah, I thought that probably a very, very elegant setup, because you can see endothelial cells in the matrix gel have this tubular formation. If you put the pericytes there with it, I wondered, because then it's truly probably a capillary bed, is it?
Yeah, like some kind of 2D matrigel studies.
Yes.
We form the tube, we put in the cells in, and if they can enhance the tube formation.
That's correct. Yeah. Thank you. That's brilliant. Thank you for your.
Then I will jump in. Okay.
Yes. Can I just sorry, can I just ask in between if people would like to, so the chat is open, please enter your questions also in between so that the audience can mix in. But Grazyna, please go ahead.
Yeah, I just can say the same. This is very beautiful study, a lot of work done. I was wondering, you nicely point out about the migration of the pericytes. And actually, I was wondering, do you know what is the distance which the pericyte can go? And why do you think when there is reoxygenation that actually they go back and there is, for example, not apoptosis and maybe then local regeneration? So I would like that you will tell us a little bit about your thought here. Why do you think that it's the reverse migration?
Yeah, that's a great question. That bothers me all the time because we don't see all of them move towards arterial. A lot of them remain in the capillary levels. So that also initiate us like the motivation to study different subtypes of pericyte. We do see those type 2, like closer to the arterial. They seem to be moved back to move to the arterial. And with the question of oxygen, we don't know, and we don't know why they move back. It's maybe because the muscularization of the arteriole is calling them to move to there, but once the remodeling is no longer existing, so these cells are no longer needed at the contractility of this vessel, so they just move back. But it's such a dynamic movement, and I'm fascinated by this process. Yeah, I don't know. That's probably something we can study in the next few years.
Yeah, because I think now coming to this, what Lan said, if you will have like a models where you could like really investigate the movement of the pericytes, that would be amazing. Another question, which you also pointed to during your discussion, is that having this two populations of pericytes, we might be careful with the therapies, because while we would like to treat one of those, let's say those which are propagating in the pulmonary hypertension, we have to be careful about the other one, which might lead to the leakage. How to do that?
Yeah, that's also a great question. Yeah, there are two populations, but their PTGF receptor beta expression levels are both high in that population, and also smooth muscle also heavily express PTGF receptor beta. If you use PTGF receptor beta inhibitor to block smooth muscle, so they could have reduction in proliferation and contractility. But if you block these type 1 pericytes in capillary levels, then the integrity of the capillary will be disrupted, and you may have some fluid leakage and destroyed capillaries. Therefore, there's no gas exchange happening at the distal level in the alveoli. So that's why I think to using PTGF receptor inhibitor, we need to be careful. I think there's currently a clinical drug, etapanab, ongoing. It's blocking the PTGF receptor beta, but it's have some side effect. I think it's because it's inhibiting pericyte at the capillary level. And the reason Vimentin, we think it's interesting, is because it's commonly upregulated both human pericyte and mouse pericyte. That's how we go to that route. But maybe pericyte become fibroblast-like cells because Vimentin is a fibroblast marker. But in order for them to move, they must have this acting organization, and Vimentin is also regulating the cytoskeleton of cell movement. So that's another reason we focus on Vimentin. So could be something in the future, Vimentin inhibitor could be effective for therapeutics.
Well, we might have a little bit cautious here because Vimentin is also high expressed in the endothelial cells, as they're also mesenchymal cells. So one could test that, but just something maybe to consider, as Vimentin is not only fibroblast, but also endothelium. You know, I always think when some cell population has some gene, it has to have a meaning. So could you actually speculate why pericytes are so enriched in this HIG-D1B? Why functionally?
So this HIG is also representing hypoxia relevant gene. So that's another reason we pick up this one. We think hypoxia is driving pericyte movement, because in my lab, we have a pneumonectomy model, which is increased blood flow, because we remove the left side of the lung lobes. We even remove one additional lobe from the mouse lung. The mouse stably developed pulmonary hypertension, but we think this is something about blood flow, shear stress induced pulmonary hypertension. So when we use this animal with pericyte label, we don't see pericyte move to the arteriole. So I think hypoxia definitely driving this cell movement. We need to tease out what's the exact mechanism. But HIG is relevant to hypoxia. That's another reason we pick up HIG. But the function of HIG, there's probably one literature talking about HIG in heart. There's nothing more about it.
It could be also very interesting, like to try maybe for your future studies to understand what actually this protein does.
Exactly.
Still a lot of work which you can continue. That's definite.
Absolutely.
Jurjan, do we have questions from the audience?
I don't have any questions in the chat yet. Samantha, can people directly
I have one question.
Yeah, go ahead.
Wenjun 和Yuryang's PhD students, hi Ke, hi every audience. Thank you very much for your excellent presentation. It's really impressive. I maybe have a simple and maybe stupid question. I like how you classify the pericytes as high or low based on the marker expression. But I'm curious about, do you think these two subtypes truly represent distinct cell types with different functions or rules in the tissues, or could they just represent the cells in different status? Can you hear me?
Yeah.
Yeah.
That's also a very good question. It's also, we're being asked that question from the reviewer. So this, when we look at the different expression levels of these genes in human cell, long cell atlas datasets, we keep ourselves really unbiased. I just want my bioinformatician look into it, and just we do very blinded cell clustering. And first of all, I think we mixed the donor sample really, really well. They don't have any batch effect. And secondly, Hong, he conducted very meticulous analysis. So we do see cluster 2 is slightly different from the others, but the p-value is less than 0.05. So I trust that data. And when we look at the expression levels of the different genes, he is able to figure out the levels of CSPG4 PDGF in these two clusters, and also we do see after hypoxia, our Hig TDT cells are moving like to, we think majority stay in capillary. There are some of them moving, so that make us confirming it is gonna be two populations. And when we look into the literature, people keep talking about subtypes of pericyte in different organs that kind of confirm our speculation. But I would agree, in human lung samples, we need to provide more evidence to show there are indeed two different types of pericytes. But you guys probably also familiar with in endothelial, the recent work on subtypes of endothelial cells. There's no validation in human lung samples. There are two types of endothelial cells. It's just based on the single cell data. There's no staining data, etc. In mouse, it's being labeled with immunofluorescence staining. I do think these subtypes of vascular cells in human lung should be more carefully examined.
Okay, thank you. Thank you so much for your explanation.
Yeah, actually, I had a kind of follow-up question on it, because in your hypoxia data, I think you did the tamoxifen injection before the hypoxia exposure, isn't it?
Yeah, so tamoxifen is also tricky. We have to I didn't input here, but we do dosing tamoxifen. So we do dose from very minimum to see if we got label to the maximum, how much we can label. We do see the maximum we label, almost 100% of the cell get labeled. There's no smooth muscle cell get labeled. And we decreasing the dosing and get the optimal dosing. But we only gave, I believe the ratio we do give is, I think, 4 milligram per 25 gram body weight of the mice. So it's not a lot tamoxifen we gave, and we let the mice to digest tamoxifen for a week. Then we conduct experiment. So anything we see in the label, it's permanent. Nothing. It should be completely digested, the tamoxifen.
Because it looks in your follow-up data, like 1 week, 2 weeks, 3 weeks, like that, the TD positive cells, they move like towards the arteries, the smooth muscle positive vessels.
Right.
And then, like phenotypically, they switch into more the type 2 cells. So that's and although it's of course not a completely a time frame experiment, but that suggests that there is a kind of a dynamic switch between the two cell types, isn't it?
I think the type 2 pericyte, we think it's more lineage active because they have lower expression of HIC but higher expression of PDGF. So and they are spatially they are closer to the arteriole. So that's why they are quickly responding to hypoxia. They move towards arteriole and become SMART positive cells. I think they become SMART positive so they could have more contractility for the vessel. But the type 1 cells, I think they remain at the capillary, but because the loss of the type 2, they need to cover more surface area of a capillary, so they grow their processes for this absence of type 2. Did I answer the question?
Yeah. I think it remains quite an intriguing issue because I think that the data still leave a possibility as a kind of a phenotypic switch. I think there is there's time for a last question because I think we are almost running out of time. So is there one last question from the audience? Otherwise, I will keep it with this, and I would take the rest of the time for thanking you, Ke, for this wonderful presentation, for lighting the discussion, because I think we had quite some discussion on a very novel topic, which actually highlights the complexity of the disease. Grazyna and Lan, thank you for the continuing the discussion, and I think driving it into a really nice scientific debate, which I appreciate a lot. So thanks, and finally, all the people who stayed in the discussion and the presentation, thanks for your attention. And looking forward to see you all in the ATS, which Samantha is already advertising in the background. So hope to see you all there. Thank you very much.
Thank you.
Thank you.
Thank
Thank
you.
you so much.
Thank you.
Thank you.
Have a good one. See you.
Very nice work. Thank you.
Excellent. Take care. Bye.
Bye-bye.